Good afternoon, everyone. My name is Whitney Hoot, and I'm really grateful to be here today and to have the opportunity to learn about reef restoration efforts here in Australia and around the world. For me, I think restoration is a source of hope. So thank you all for being here today and sharing your knowledge and experiences. I think everyone knows that there are major obstacles to restoration, scalability being perhaps the most daunting challenge. But I've been really inspired by the talks here over the past couple of days, and I think we're moving forward to making restoration a meaningful tool to rehabilitate degraded reefs. So Guam is a U.S. territory in the western tropical Pacific. It's about 2,500 kilometers from the Philippines. If you don't know where Guam is, don't feel bad. Most Americans don't know where it is, and it's a part of our country. The island of Guam has an area of about 540 square kilometers, and we've got about 110 square kilometers of coral reef area. Our reefs are quite diverse. We've got over 5,000 reef species, including about 350 species of scleractinian corals and about 1,000 fishes. We have a population of about 150,000 people, but we do get over 1.5 million tourists every year, which obviously puts extra stress on our reefs. The indigenous people of Guam are the Chamorro people, and they make up about 37% of the population. Guam's reefs have been impacted by widespread bleaching 4 out of the last 5 years. And as far as we know, this is an unprecedented frequency of severe bleaching globally. So this makes our reefs tragically unique. We've also been impacted by both acute and chronic outbreaks of COTS in recent years. So with these stressors and with other local impacts, such as land-based sources of pollution and extensive overfishing, coral cover on our seaward slopes has declined by about 80% in the last 50 years. We had a break from coral bleaching in 2015, but unfortunately our reef flats were heavily impacted by extreme low tides event related to ENSO. And due to the combined impacts of bleaching and exposure, we lost about half of our Staghorn Acropora coral between 2013 and 2015. And this is equivalent to about 17.5 hectares of coral cover. So to date, most of our restoration efforts on Guam have focused on staghorns. They're a vital component of our reef flat communities, providing essential fish habitat and nursery grounds, protecting coastal infrastructure, facilitating traditional practices, including cultural fishing methods, and supporting our growing tourism industry. And tourism is the largest sector of our economy. About 1/3 of all jobs on Guam are in tourism, and our reefs are visited by about 300,000 snorkelers and 100,000 divers from off-island every year. We have 21 known populations of staghorn corals of 6 species on Guam, or we had. On this map, so the size of each circle is relative to the area of the staghorn assemblage at this site. Between 2013 and 2017, 4 of these populations had 100% mortality. And 4 additional populations experienced 80% or higher mortality. We have almost no surviving staghorns left on the east side of the island, and we're obviously really concerned about the loss of these corals and their associated ecosystem services. This sudden, severe, and widespread degradation of our reefs due to both climate change and local stressors has stimulated significant interest among local coral reef managers and scientists in coral reef restoration. And we think that without urgent, immediate intervention, we're likely going to continue to lose genetic diversity of corals and potentially face local extinctions. So one of the main obstacles of successful reef restoration and one of the primary criticisms of restoration efforts is the challenge of restoring degraded reefs that exist in suboptimal conditions due to warming and local stressors. So successful restoration has to be strategic and data-driven, and we have to take into consideration best practices for site selection, species selection, and restoration techniques, while incorporating findings from monitoring on mortality, recovery, and also physical factors. Appropriate site selection is absolutely crucial for successful restoration, and it has to be based on a range of abiotic, biotic, and also socioeconomic considerations. And as we know from this session, water flow is extremely important in site selection for both in situ nurseries and also outplant sites. High water flow regimes have been seen to provide refugia from warming, which can increase coral reef resilience and thus safeguard the success of restoration efforts. When you have high water velocity over the surface of a coral, you have increased nutrient uptake by the zooxanthellae, which stimulates symbiont growth, tissue repair, reproduction, photosynthesis, and respiration. For the coral hosts, high water flow rates can facilitate heterotrophic feeding by increasing both prey encounter and capture rates. And heterotrophic feeding can reduce thermal stress and ameliorate bleaching, as some coral species can compensate for the lost nutrition from the loss of their, the photosynthesis of their symbionts by filter feeding. So elevated light and water temperatures cause chronic photoinhibition in the coral symbionts, and this leads to a buildup of reactive oxygen species within the coral tissues. This is theorized to induce bleaching as the corals expel their zooxanthellae once the reactive oxygen species begin to damage the host cells. So high water flow can actually mitigate heat stress and bleaching by increasing gas flux at the surface of the coral and the boundary layer with the water, and diffusing these reactive oxygen species. A recent study on Guam, we found a significant positive correlation between the distance from the reef margin, or the reef crest, and coral bleaching severity among staghorns. So what we found is that offshore staghorns bleached less and had lower mortality, which we hypothesized to be driven by increased water flow due to wave action and flushing near the reef margin. This is a project being led by James Fiferb, who's a graduate student at the University of Guam. I am not a molecular ecologist, so excuse me while I give you a non-technical explanation of his work. So we conducted both in situ and lab-based experiments to determine whether water flow can impact gene expression in heat-stressed corals. For the in situ experiment, we sampled Acropora pulchra colonies from a high flow and a low flow area at one reef flat site in August 2017, which was at the tail end of our most severe coral bleaching event to date on Guam. Using clot cards, we determined that the flow rate at the high flow site was about twice that than at the low flow site, and there was no significant difference in temperature between these two areas at this site. Through an ex situ experiment, we examined how water flow regime influences gene expression in acutely heat-stressed corals under controlled conditions. We sampled corals from the high flow site, and we allowed them to acclimate in the lab in ambient temperatures in 4 unidirectional flow tanks. After 11 days of acclimation, we split each tank into high flow and low flow sections, and then we exposed 2 of the tanks to acutely elevated temperature, and this was designed to simulate the heat stress that's experienced during a coral bleaching event. Samples of each fragment were then gathered after this simulated heat stress event and sequenced for gene expression analysis. So what we found is that water flow regime can produce a transcriptomic response, which is evidenced by the differential gene expression. So we found a significant variation between high and low flow samples at ambient temperature in the ex situ experiment, and between the high and low flow samples that were collected from the reef during the period of thermal stress. We then compared the low flow samples, which are shown on this graph in brown, and the high flow samples, which are shown in green. against the heat stress samples from the lab-based experiment. And we performed blast-to-go analysis on all of the differentially expressed genes. You can see here all of the differentially expressed genes that are related to heat stress. So these are heat stress candidate genes. Bars to the left of the axis indicate that the gene is downregulated within the ambient temperature conditions compared to the heat stress corals, and bars to the right show that the gene is upregulated. So this indicates that for the high flow samples, which are shown in green, that they are less downregulated compared to the heat stress samples than the low flow samples are when compared to the heat stress samples. So this is a phenomenon called front loading, and this is the upregulation of stress-related genes at baseline conditions. So in the high flow samples, the expression of the heat stress genes is more comparable to the heat stress samples than it is in the low flow samples. So essentially, the corals are prepared at baseline conditions for heat stress when they're in these high flow conditions. Other studies have shown that front loading is characteristic of resilient corals. So corals with front-loaded heat stress genes may be able to respond faster to stressors and thus acclimate better and have higher recovery. We suspect that the increased resilience to bleaching, which is evident among corals in high water flow environments, may be due to this front-loading effect. So based on this work and other studies, we have three possible implications of water flow conditions on restoration success on Guam. So first, nurseries and outplanted populations may have greater bleaching resilience and survival if they are situated at sites with high water flow regimes. Secondly, coral fragments that are collected from donor colonies in high flow regimes and then outplanted directly onto reefs, or corals that are transplanted into nurseries that have high flow regimes, may demonstrate this front-loading effect and thus be more resilient to coral bleaching. And lastly, something that we found is that water flow rates should be considered when you're designing nursery structures and choosing outplanting methods. Because another result of our study is we found there can be considerable variability even within colonies or within staghorn patches for water flow. So the reason why we initially conducted this experiment is because we anecdotally noticed that the outsides of the staghorn patches were bleaching less and the interiors were bleaching more, and we suspected that was due to water flow. So in a coral nursery, it's possible that coral fragments that are situated on the interior of the nursery may be less resilient to bleaching if they're getting less water flow, or when you're planting your fragments onto the reef or settlement tiles, if they're blocked by coral colonies or other structures in the water, they may not have sufficient water flow and their resilience may be impeded. I lost a photo. So on Guam, we are incorporating this work and other research as we upscale our restoration efforts. We recently completed our first large-ish scale outplanting on Guam. This was in the Tupungan Channel, and we outplanted about 300 fragments of staghorn corals, Acropra pulchra, into 3 25 square meter plots. And one of the reasons why this site was selected was, one, because of depth, and two, because it has good water flow. We're also experimentally doing microfragmenting with massive corals, and we've also transplanted some clonal fragments of a couple different Acropora species onto stable A-frames to hopefully create spawning stocks. We have one coral nursery right now that has about 1,000 fragments of 5 species of staghorn coral, and we're looking to transplant more massive corals and other morphologies into the nursery. We're also in the process of site selection for a second coral nursery, and water flow is a big consideration in that. Additionally, we've done some work with sexual propagation, and we have used both culturing in the lab and also using in situ settlement pools and then with settlement tiles inside of them. And that was work done with Seacore International. On Guam, we recently founded a new group called the Guam Reef Restoration and Intervention Partnership. And the main objective of this group is to develop an island-wide coral reef restoration strategy. So we want our efforts to be strategic and data-driven and cohesive. And it sounds like just like in Australia, we have some strong critics of restoration in our local scientific community, which can make things difficult, particularly when you have such a small group of people working on coral reef research and management. We aim to have this document prepared by the end of the year, so hopefully we'll have a solid plan moving forward. We also hope to do restoration of other systems, including mangroves and seagrasses, and another primary goal we have is community engagement. Without buy-in from the local community, we will not be successful in our efforts. And we have worked on developing an Adopt a Reef program. Right now, we have local communities that have adopted reefs, and they go out and they do coral reef monitoring in this reef. But we would like to branch out and have communities actually do outplanting on their local reefs and then monitor and manage these outplanted populations. Thank you very much.